A very good morning, children. How are you all? Hope you have understood the previous sessions of the plasma membrane. Okay, if you have not understood any topic or if you left out some topics and all, you can go ahead with the videos already which are there, and the link I have given in the description box. You can look at that. Okay, so today we are going to see an activity where we will understand how the materials move into the cell and move out of the cell. The same thing which you have learned yesterday, that is entry of the materials into the cell and entry of the materials outside the cell. To understand this better, this statement better, what we are doing here is we are doing a very small experiment here. Okay, you can see this apparatus, the way it is arranged. Okay, here you can see a retort stand. Okay, and we have attached a funnel to that, and then we have taken a beaker here. Okay, apart from that, what we have taken here is we have taken a small beaker, okay, which is filled with 100 ml of water, and also what we'll do is we'll add 1 tablespoon of rice powder or wheat powder. So what we are adding here, we are adding either rice powder or wheat powder, so to that water. And also what we'll add is we'll add 1 drop of iodine solution. We will add 1 drop of iodine solution to it. So after adding 1 drop of iodine solution, now what we'll do is we will take this beaker, okay, as I have shown in the figure. What we will do is we'll pour this contents into the funnel, and now before pouring that into the funnel, what we'll do is we'll put a filter paper over here. Okay, this green color one which you can see here is a filter paper, and I have taken the filter paper here and I kept that in the funnel. Okay, and then what I am doing is I am pouring this all contents into the funnel, so that, and also I have kept the opening in such a way that this contents will fall into the beaker. Now when you pour them into the beaker, what are the things which are coming into the beaker? What are the things which comes into the beaker? Now what's happening is the water which is dissolved, the water basically what is there, so that will filter into the that will pass the filter paper and they'll come into the beaker. Okay, first one is water. Second one is what are the things which are soluble in the water, the things which are dissolved in the water. So what are the wheat flour or the rice flour which is dissolved in the water will also come into the beaker. So whatever the undissolved thing is there, the thing which is not dissolved, which is undissolved, the wheat flour which is completely undissolved will stay back in the filter paper as a precipitate. It will stay back in the filter paper as a precipitate, so that will not be filtered. So whatever the things which are receiving here is the water and also dissolved rice flour. That means this filter paper is not allowing everything to pass through that. So from this experiment, what we are able to understand is it is allowing the water to pass through it, and also it is allowing the dissolved flour, dissolved rice powder or dissolved wheat powder to pass through it. It is not allowing any other thing else. Okay, now in the same way, cell membrane also acts in the same way. Even if you take the cell membrane, it also acts in the same way. This cell membrane will allow the water to pass through it. Second one is it will also allow the dissolved materials or substances to pass through it. It will not allow the undissolved things to pass through it. Okay, that is what is done by the cell membrane also. So if the cell membrane is not allowing such kind of things, that is, there are some materials which are not allowed by the cell membrane to pass through it, and there are some substances which are being allowed to come into the cell. So those kind of that is a condition, okay, this particular condition of the cell membrane where it is allowing few substances to pass through it and few it is not allowing them to pass. This condition is called as permeability. Very important term, remember this. That means our cell membrane has permeability. What is permeability here? It allows only few or certain substances which are useful to that will to pass through into it, and what are the ways are there that will be removed out. So allowing only certain substances to pass through it is called as permeability. It's called as permeability. So to know more about the plasma membrane or the cell membrane, okay, we'll go ahead with the few things about the cell membrane. Now if you see the cell membrane, okay, so if you see the cell membrane, there are some words which describe the cell membrane. The first one is, it is extremely delicate. It is extremely delicate. The first word which we are using to describe the cell membrane is, it is absolutely delicate, and also it is elastic. It is also elastic, and it is a thin membrane. It is a thin membrane which covers the cell. Now if this is the cell, this is the inner parts of the cell, and what you can find in the cell, you can find the cytoplasm and also nucleus and all the other organelles. So all these organelles, they are not able to come out because they are covered by a membrane which is called as the plasma membrane. So this plasma membrane will act as a protective sheet around the cell. That is the first point. Okay, so this cytoplasm, this cell membrane will divide this cell from the other cell. So now if there is another cell which is beside it, this plasma membrane is dividing this cell from the other cell. Okay, so now this cell membrane will not allow anything to pass from this cell, and it will be very selective. Only what are the things which are required for this cell, they will allow them to pass inside. So it is very selective. So we also call this plasma membrane as selectively permeable membrane. We also call it as selectively permeable membrane. So, and in the other words, you can say that it is the living flexible membrane. So this plasma membrane is delicate, at the same time is elastic, and it is a single membrane, thin membrane, which is covering the entire cell and protecting the parts which are internal parts, that is cytoplasm and the nucleus and also other organelles. It is dividing this cell from the other cell, and it allows only few substances, few selective substances to pass through it. Okay, that's the reason why this cell membrane or plasma membrane is also called as selectively permeable membrane. It is also called as living flexible membrane. Is it clear, children? This one? Okay, great. Okay, children, so here we are going to see some functions of the cell membrane. Children, here are going to see some of the functions of the cell membrane. So the first function of the cell membrane is shape. It gives a definite shape to the cell. So in all the other 2 units we have studied that each and every cell is not in the same shape and size, isn't it? Each and every cell has its own shape. Different cells which are present in the different tissues will have their own shape, isn't it? So that's why we can say that this particular plasma membrane will give a definite shape to the particular cell. Okay, already we have seen that it is not a 2D but it's a 3-dimensional So it gives a shape to the cell. Second one we have seen is the mechanical barrier. So the mechanical barrier in the sense it will protect the internal contents of the cell. It will not allow any of the internal contents to pass out. So it is acting as a barrier. It is acting as a barrier. So it is actually stopping the things from entering outside. The inner material will not be able to enter outside. It's like a gate which is closed, which is locked, so that nobody from inside can go outside. So it is acting as a barrier. In the same way, we have learned about the semi permeability or selectively permeable membrane. So here selective permeability means the same thing. So whatever the substance are there, it is selective, very selective in allowing them inside, and is very selective from allowing them to go outside. And the same like it's a corona time, so we are very much selective whom to enter inside, whom to allow people to come inside, whom we should not allow to come inside. So if we do, that is a kind of selective. We are very, very, very selective, and also we are very selective for which works we have to go outside, for which works we need not go outside. We are selective. In the same way, here the cell is also, the cell plasma membrane is also, or the cell membrane is also selective, because it is selecting only few things to pass inside and not allowing all the things to pass inside or to go outside. So that is the selective permeability. Next one is the endocytosis. So this endocytosis, the best example what you can take is the amoeba. Amoeba is the best example which you can take here. Amoeba is only a single celled organism, unicellular organism, which has only plasma membrane covering its body. So now if we want to eat food, it cannot eat the food as we are eating. We have hands and all, we can take the food. So it cannot do that. It has to take the food through its cell membrane. So the cell membrane allows few things or foreign substances to be engulfed into the cell membrane, through the cell membrane into the cell. So that is how they will take the organism through the cell membrane or through the plasma membrane into the body, into its one single cell. So this is called as the endocytosis. So this same endocytosis is also seen in some of our WBC, where they will engulf the microorganism, which we have seen in the blood cells. Okay, and next one is the recognition. Next one is the recognition. If any foreign particles enters inside, okay, so our cell membranes are very good in recognizing them and immediately stops them, or it will act as a barrier. It stops them, and immediately it will allow some of the cells to come inside, or it will send information so that they will allow the antibodies to come out, and these antibodies will come and they will kill the foreign materials, or they will neutralize the action of the foreign materials. So that is how they will recognize the things. And next is flow of information. So from one cell to the other cell, if the information has to be given from the one cell to the other cell, so even the plasma membrane will do that. So they will do in the form of, they will take the help of the RNA, that is the ribose nucleic acid. So that is what they will help in the flow of information from one cell to the other cell. Next is the osmosis. We have seen already, moving of the water from the lower concentration to the higher concentrations. That is what we call as the osmosis. So there are some special channels for the osmosis, so that in the plasma membrane especially, so that water can travel from one cell to the other cell. So that is how the water will move from the lower concentration to the higher concentration through a process called as osmosis. And the next one is the cell continuity. Cell continuity. So now there are some cells. This is one of the cell and this is one of the cells. So this is the adjacent cell. Now here you can see there are two cells which are attached here, and there is one more cell which is attached here. So there are several attachments, right? So now even though this cell is covered by plasma membrane, okay, and again this is also covered by the plasma membrane, there are some separate channels of opening into the other cells, so that they can give the information and also they can send the materials, so that there is continuity of the cells. The cell is not a completely closed structure. Okay, they allow few substances to pass through them, so they will continue with the other cells like this. So that is how we can see the continuity of the cells. And then there is specialization. What is specialization? So if you have seen the small intestines, they are coiled structures, isn't it? So like this, there are coiled structures which are called as small intestines, and in few places the small intestine is modified into finger-like projections which are called as microvilli. Okay, the plasma membrane here, they forms some structures called as microvilli, and all these microvilli structures will absorb the food material and pass them into the blood. So that is how the specialization works here. So these are the different functions of the cell membrane. First one, they give shape to the body. Second one, they act as the mechanical barrier. Third one is they are selectively permeable, and fourth one, they show the endocytes engulfing the foreign materials. Fifth one is the recognition of the foreign materials and also release the antibodies so that they can be killed or they can be neutralized in their actions. Next is flow of information from one cell to the other cell, and next is the osmosis, that is from lower concentration to the higher concentration, and next is the cell continuity with the help of the plasmodesmata, and last one is the specialization, like structures called as, you can see here they are called as the microvilli. So these are the different functions of the plasma membrane or the cell membrane. So the cell membrane in the cell will perform these many functions. So the major function here we have learned is the selective permeability through all the other examples or the experiments which we have done. Is it clear? So the next what we are going to see is the transport across the membrane. Okay, the exit or the entry of the transport, or you can say the entry and exit of the materials through the membrane. So what are allowed and what are not allowed? Okay, basing on that, this cell membrane can be of four different in different four conditions, you can say. In the first condition, so if solvent and solute both are allowed, then we call this cell membrane as permeable membrane. It is permeable. That means it is allowing both solvents and solutes into the membrane. So we call it as the permeable membrane. So if solvent and solute, if they are not allowed, or any other substances, if the cell membrane is not allowing them to pass through it, then it is called as the impermeable. It is called as the impermeable membrane. So in first condition, it allows the solute and also solvents to pass through them. That's why it is called as the permeable membrane. In the second condition, if it is not allowing the solvents or solutes to any other substances to pass through them, that's why this condition is called as the impermeability or impermeable membrane, you can say. The third one is if it allows solvents but will not allow the solutes. All the solid particles will be left away, and what is left, only the water. If it is taking the water, that means it is showing what? It is showing semi-permeability. That is, now this membrane will be called as semi-permeable membrane. So that means if it is allowing only solvents but not solutes, that's why we call it as the semi-permeable membrane. And in fourth condition, if it is allowing specific solutes to come inside, so if it is very much specific or if it is very selective in allowing the things to come inside, then we call it as the selectively permeable membrane. So this is how we can divide the selective or you call the cell membrane into four different kinds basing on its actions. The first one, if it allows the solvent and solute to enter, we call it as permeable. and if the solute and the solute and if the solute and the solvent are not allowed, or any other substances are not allowed to pass through them, then it is called as impermeable. Okay, and then if it allows solvents and not solutes, so it is not allowing the solutes to come inside, but it's allowing the solvents to come inside, then it is semi-permeable in nature. And then if only specific solutes, if they are very selective, only these solutes should pass. So if it has that kind of condition, then it is called as selectively permeable. So this is how the cell membrane can show these kind of variations, these kind of actions. Okay, so by this we come to the end of this today's topic, and okay, until then, see you all, have a great day, take care.